Today I am going to discuss about the DNA replication. So it is basically the formation of new DNA from previous DNA. So one thing. So next thing is DNA has two strands, and these two strands are anti-parallel to each other, means the three end of the one DNA strand faces the five end of the another DNA strand. So both of these two strands are anti-parallel to each other. So another thing, DNA polymerase, which is the main enzyme involved in the DNA replication, synthesize DNA only into five to three direction. So in this the way, the one strand of DNA synthesized continuously and another strand synthesized in segments. So the DNA strand which synthesized continuously is known as leading strand, and which is synthesized in segments is known as lagging strands. The origin of replication is basically the certain sequence of nucleotide, which is 80 rich sequence, adenine and thymine. So the next important things, helicase is an enzyme. This helicase which break the hydrogen bond between the adenine and thymine. Adenine and thymine has two hydrogen bonds. So helicase break these hydrogen bonds. So next step is the topoisomerase. Topoisomerase is basically the class of the enzyme which prevent the supercoiling in the DNA when the DNA is unwinded by the helicase enzyme. Helicase is an enzyme which open the DNA for DNA replication by breaking these hydrogen bonds. So the topoisomerase is a class of enzyme that prevent the supercoiling of the DNA. One thing. Second, there are the many types of the DNA topoisomerase. In eukaryotic, this is super high yield stuff. In eukaryotic, topoisomerase 1 is inhibited by anti-cancer drugs. For example, is topotecan. Topotecan is anti-cancer drug, and DNA uh, topoisomerase 2 in eukaryotes is inhibited by adipocyte drug. So these are the anti-cancer drugs, and another clinical significance of these DNA topoisomerase enzyme in prokaryotes, it is inhibited by fluoroquinolone. Fluoroquinolone is a class of antibiotic. So DNA topoisomerase is a super high yield stuff for the USMLE point of view. Now I am going to discuss about the DNA replication in detail. So in DNA replication, helicase enzyme unwind the DNA by breaking the hydrogen bonds between the adenine and thymine nucleotides. So, so helicase open the DNA by breaking the hydrogen bonds. And then what is the function of the polymerase? Polymerase form phosphodiester bonds, and nucleases hydrolyze the phosphodiester bond. Now, after opening the two DNA strand by the helicase, an enzyme known as a primase, this enzyme form RNA RNA primer, which is basically the nucleotide sequence of the RNA. So DNA polymerase does not initiate itself the DNA replication. So it's work on RNA primer. This RNA primer, the RNA nucleotides are removed by another enzyme, and after that, this RNA primer is elongated by the DNA polymerase. So there are the two strand. One is a leading strand and another is a lagging strand. So basically, DNA has two strands. Parent DNA. This strand is this is a three end of the parent strand. So the new DNA polymerase has a characteristic. It synthesizes DNA in a 5 to 3 end direction. So this DNA 5 to 3 end direction synthesize continuously, while this DNA, this is the parent strands. So, DNA synthesized in this way. So the segments of the DNA are synthesized in the lagging strand, which are known as the Okazaki fragment. And these strands are later on joined by the ligase enzyme. So one another thing, after opening of the DNA, unwinding of the DNA by this helicase enzyme, then this is immediately the open area of the DNA strands are binded by single stranded binding protein, because if this is a DNA open strand is exposed by these nucleases enzyme, so they hydrolyze these phosphodiester bond and the degradation of the DNA take place. So this is a basically the explanation of the DNA, how DNA replication take place. We can divide the DNA repair into two types. One is a double-stranded repair and second is a single-strand repair. So in double-stranded repair is further divided into the two type: non-homologous end joining and homologous double-stranded DNA repair. So what happened in the non-homologous end joining repair of the DNA? Here is the two ends of the DNA fragments are brings together and joined. Then if this repair does not take place, there are condition known as ataxia telangiectasia. So this is a super high yield step. So ataxia telangiectasia is a basically the example of non-homologous and joining defect. So next is a homologous recombination double stranded repair of the DNA. Here it requires two homologous DNA strands. So two homologous DNA duplexes are required. A strand from the damaged DNA is repaired by using the complementary strand from the intact homologous DNA. So if this kind of repair does not take place, then this is lead to the breast and ovarian cancer. Breast and ovarian cancer. So where the genes are BRCA1 and 2. And it also happen in Fanconi anemia. So again, the clinical significance of the non-homologous end joining is ataxia telangiectasia, which is super high yield stuff. Second things, if the homologous recombination DNA repair does not take place, then this leads to the breast ovarian cancer, BRCA1 and 2, and Fanconi anemia. So this homologous recombination restore DNA accurately, restore DNA accurately, and there is no loss of the nucleotides. There is no loss of nucleotides, no loss of nucleotide. While in case of the non-homologous end joining, loss of nucleotide take place. So this is not a accurate, not accurate because there is a loss of nucleotides. Now we are moving toward the single strand of the DNA repair. In single strand of the DNA repair, there are the three types of the single strand DNA repair. One is the nucleotide excision repair, second one is the base excision repair, and third one is a mismatch repair. So what happened in the nucleotide excision repairs? In nucleotide excision repair, UV exposure lead to the formation of the pyrimidine dimer. Suppose this is a thymidine dimer, and this thymidine dimer is removed by the endonuclease, and then newly replaced segment is formed. If this kind of the repair does not take place, then this lead to the XP disease, which is a xeroderma pigmentosum. So what happen in a xeroderma pigmentosum? These thymine dimer are not removed and replaced. So the endonuclease activity, endonucleases are deficient in the xeroderma pigmentosum, and the patient present with dry skin, photosensitivity, and skin cancer. So this is a super high yield stuff. Xeroderma pigmentosum is a super high yield stuff. So xeroderma pigmentosum. Next is the base excision repair. In base excision repair, the base is damaged. Here is the base is damaged, deaminated, and then there is a glycosylase. Remove this damaged base. This damaged base is removed by the glycosylase enzyme, and there is a AP site formed, and this is a AP site. Again, then the endonuclease, AP endonuclease and lyase. So AP endonuclease remove the five or cleave the five end, while the lyase cleave three end. So AP endonuclease cleave five end and lyase cleave three end. And this base excision repair is take place. So this kind of the repair occur throughout the cell cycle. Base excision repair occur throughout the cell cycle, and it takes place in spontaneous, spontaneous and toxic deamination and toxic deamination. Now the last one is the mismatch repair. So mismatch repair is a basically the nucleotides are mismatched. Here is the nucleotides are mismatched because G are bond with cytosine, but here is the guanosine formed with adenine. So this is a mismatch. So this mismatch segment is removed and the new segment is formed. So there's the clinical significance. This mismatch repair take place in a S phase of the cell cycle. So if this type of the repair does not take place, then this lead to the Lynch syndrome, which we'll discuss in detail. Lynch syndrome is also known as HNPCC. H stand for hereditary, N stand for non-polyposis, and non-polyposis, and C for colorectal cancer. So this is the basically the superficial overview of the DNA repair. So mutation is basically the permanent change in DNA. So it is basically the permanent change. There are many type of the mutation. One of the type is a point mutation. So what happened in the point mutation? Point mutation basically one in which one nucleotide is added, may be replaced or removed. So next is the frame shift mutation. So what happened in frame shift mutation, in which nucleotides are deleted or inserted, which is not dividable or divisible by number 3, which leading to the misreading of downstream nucleotides. So now is a splice site mutation. So what happens in splice site mutation? It is a basically the retained introns in messenger RNA. So now this is the basically the normal picture. So this is the coding strand, this is amino acid, and this is a genetic code. So the genetic code is basically the triplet sequence of the nucleotides. So UAA represent the leucine amino acid, and this genetic code is determined by the TTA nucleotide triplet on the DNA. So in what happened in the silent mutation, this G triplet G only one G guanosine nucleotide is uh, uh, replaced, and the what happened in the silent mutation. So sorry, basically that this sequence is replaced, and what happened, the is again replaced by this genetic code, again represent the leucine amino acid. So next is the missense mutation. So what happened in the missense mutation? So the basically the this middle cytosine is uh, take place of the thiamine. W- So what happened when this thiamine is replaced by cytosine? The genetic code is represented, and this genetic code represent another amino acid, which is abnormal. So example of the missense mutation is a sickle cell anemia, where the glutamate is replaced by the valine amino acid at one place. Next is the nonsense mutation. So what happened in the nonsense mutation? The only one nucleotide is replaced, and what happened, the genetic code become the UG, and this UGA code represent the stop codon. It is a stop codon, it does not represent any amino acid. So the protein produced by this type of mutation is a shorter than the normal protein. So example of the nonsense mutation is hemophilia, where the factor 8 is deficient. So next is a frame shift mutation. As we already know that the frame shift mutation, which is basically the addition or deletion of the nucleotide that is not divisible by 3, and which leading to the misreading of the downstream nucleotides. So what happened in deletion that causing frame shift? Example are the Duchenne muscular dystrophy, where the dystrophin protein is decreased or uh, dysfunctional, and insertion causing a frame shift are the Tay-Sachs disease, where the hexosaminidase enzyme is decreased or non-functional, and the deletion not causing frame shift, that is a cystic fibrosis, and insertion not causing frame shift Huntington, because these are the multiples of 3. Insertion or deletion that not causing frame shift, because they are the multiple or divisible by number 3.